All right, today we're going to talk about enzymes. And by the time you're done watching this video, I want you to be able to answer these questions. What determines where the equilibrium of a reaction will be? What determines how fast a reaction will reach that equilibrium? What do enzymes do and how? What can enzymes not do? Why is heat a poor way for cells to increase reaction rates? And what happens when an enzyme is no longer in their optimal conditions? Now, we need to talk about two terms. We've talked about exergonic reactions. We've talked about reactions reaching equilibrium, right? So a reaction is going to run until the energy of the reactants is equal to the energy of the products. Now, which direction it has to run to get there is dependent upon the delta G. Now, how fast it's going to reach equilibrium, it has nothing to do with delta G. It has to do with the reaction rate, okay? So the delta G determines which direction that reaction must run to reach equilibrium, and the reaction rate determines how fast that is going to occur. Now, we have a reaction. Take wood, take oxygen, you combust that wood with the oxygen, and it's now on fire. This reaction has an extremely negative delta G. But the questions then are, well, why aren't houses always erupting in flame? Why isn't your paper sitting on your desk always erupting in flame? And the answer is that although this is an extremely exergonic reaction, that it has to run in the forward direction quite a bit to reach equilibrium, it doesn't do so very fast. In fact, it does so very, 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 very slow. So in order to understand the rate of a reaction, how fast a reaction reaches equilibrium, or how fast, how much product is produced per unit time, we can no longer just look at the energy of the reactants and just look at the energy of products, because all that tells us is whether the reaction is endergonic or exergonic. And remember, if the energy of the products is lower than the energy of the reactants, the reaction is exergonic and will proceed with the release of energy in the forward direction. But if we want to take a look at the reaction rate, we need to look at something new. We need to look at the energy of the transition state. Now, the transition state is the state that the reactants must reach before they can be turned into products. So it's all kinds of funky ionic states and bond flexing and so on and so forth. And the important point here is the transition state always has higher energy than the reactants. And the difference between the energy of the transition state and the energy of the reactants, that is called the activation energy. So again, the activation energy is the energy necessary to convert the reactants into their transition state. All right, so this bump right here, that is the activation energy, EA. And so what has to happen is before we can get these reactants to turn into products, we have to overcome this hump. We have to invest energy. And the reason I say we have to invest energy is because, look, we get it right back with returns. And now those returns, that's the energy, that's our delta G. Okay, so the thing that is keeping our reactions slow from proceeding quickly is the activation energy, the energy necessary to reach the transition state from the reactants. Now, activation energy is a good thing, okay? There are a lot of biochemical reactions out there that are extremely exergonic, breaking down proteins into their constituent amino acids, breaking down DNA into the constituent nucleotides. Those reactions are very exergonic. We don't want them happening, okay? So, thankfully, those reactions have transition states. That the only way under physiological conditions those transition states are going to be reached is via an enzyme. Okay, and we'll get to that in a minute. So otherwise, if there's nothing, no input of energy, investment of energy to get those reactions to reach the transition state, that reaction is going to be infinitely slow. And that's good. Okay, that's good because that keeps our protein stable, that keeps our DNA stable, and we are alive. But of course, there is exergonic reactions that we do want to occur within the cell. So what does a cell do when, hey, I want to get this reaction going. I want to speed it up. We need it to go. Well, there's a couple different ways, right? One way is a maybe hypothetical way, because it's actually not a way cells use, but hypothetically, we can use heat, right? Heat would allow, that would be the input, that heat would be the energy that would allow those reactants to reach that transition state and then giving rise to the products. But heat is a horrible strategy for cells for two reasons. And the first is that when you heat up cells, you are going to potentially denature all of the proteins and biological molecules in that cell. So, number one, bad because you don't want to heat up cells because you're going to denature all your proteins. Number two is heat specific. Let's say we only wanted reactions A, B, and C, which are exergonic. We only wanted to get them sped up, but we don't want reactions X, Y, and Z, which are also exergonic. We don't want them sped up. Heat can't do that. Heat can't be specific. You heat up a cell and all of the exergonic reactions are going to be sped up. So that's not a good way. Instead, what we use is an enzyme. Now, enzymes are usually proteins, but they are sometimes RNA molecules, but most of them are proteins. And what they do is this. Let's take a look at this graph. So this is the same thing. We have our reactants, and we have the energy of our products, right? On the y-axis is free energy. And so we know that the energy of our reactants is greater than the energy of our products. And we're going to run this reaction of converting reactants to products in two different ways. One, in the absence of an enzyme, and that's black. And then two, in the presence of an enzyme specific for the reaction, and that's in red. So let's take a look. I've got a bunch of important questions. I am going to take a pause, a moment here, and I want you to think about them, and then I'll come back and we're going to address them together, because this is a beautiful example of how you can use data to really glean some insight into what an enzyme can and cannot do. So I'll pause, and now we're back. All right, so do enzymes affect the delta G? Let's take a look. Delta G is the product, the energy of the products minus the energy of the reactants. Notice whether we've got enzyme or not, the energy of the products and the energy of the reactants don't change, which means the delta G doesn't change. Excellent. Do enzymes affect a reaction's equilibrium? Remember, reaching an equilibrium is dependent upon the energy of the reactants and the energy of the products, and that reaction running until the energy of those two are equal to each other. If an enzyme doesn't affect the energy of the reactants and the product, it's not going to affect equilibrium. Next, can enzymes make an exergonic reaction or an endergonic reaction exergonic? Well, let's think about that. In order to make an endergonic exergonic, we need to increase the energy of the reactants or decrease the energy of the products. We just got finished saying that enzymes can't mess around with the energy of the reactants, and enzymes don't affect the energy of the products, and therefore enzymes cannot convert an endergonic reaction into an exergonic reaction. And finally, what determines which direction an enzyme catalyzed reaction will run? Well, what determines which way a reaction will run? Again, the delta G. Reactions always run towards the lower energy. If the products had the lower energy, then we run in that direction. If the reactants have a lower energy, we run in that direction. Enzymes don't affect directionality, and so an enzyme will catalyze whatever direction that reaction's delta G is telling us that reaction is going to run. Right. So if an enzyme doesn't do any of that, what does it do? Well, it's simple. We can see right here. It lowers the activation energy. It stabilizes the transition state of the reactants, as we will soon see. And when you stabilize something, you lower its energy. And now that the amount of energy necessary to reach the transition state has been lower, it will be easier for the reactants to have energy invested into that. And now that reaction will be faster. That is what an enzyme does. Now, enzymes most of the time are proteins, and they have a structure like this with a cleft in which a substrate binds. And that is called the active site, or sometimes called the catalytic site. Now, how do enzymes interact with their substrates? Well, they interact very specifically. So there's only one or a couple different substrates that will bind to any given enzyme. And they interact through weak interactions: hydrogen bonding, hydrophobic interactions, ionic interactions. Now, the way in which an enzyme catalyzes a reaction is called the catalytic cycle, where the substrates bind to the enzyme, and now we have an enzyme substrate complex there. Then catalysis occurs, so we convert those substrates to product. And now that product is released, and that enzyme is able to catalyze another reaction. Now, we call these substrates reactants when we are talking about enzyme catalyzed reactions. Reactants we refer to as substrates. And don't forget that depending upon the delta G, the enzyme can catalyze the reverse of this direction, where the enzyme can bind to the products and convert the products to the reactants and release them there. So it depends upon the delta G, but enzymes can catalyze both directions of a reaction. Now, again, I've alluded to this a little bit before, but let's talk about it officially now. How do enzymes lower activation energy? They do so by stabilizing the transition state. Okay, here we have the enzyme in blue, and we have the substrate in our bar. And let's say the reaction here is we are trying to snap this metal bar in half, and the transition state is to first bend it. Well, the enzyme has evolved not to stabilize just the natural state of the substrate, the straight bar. Instead, it stabilizes the transition state of the substrate. So now it's easier to bend that bar. The bent bar has been stabilized. And in so doing, we therefore lower the energy of the bent state of this bar, the transition state of the substrate. And what are the sorts of factors that allow it to stabilize? Well, it provides a favorable microenvironment to the transition state. The appropriate pH, the appropriate ionic conditions. It orients the substrate such that the two parts that need to react to each other automatically are facing each other when they bind properly to the enzyme. So all of these factors go into allowing that enzyme to properly stabilize the transition state of the reactant, of the substrate. Now, enzymes evolved under specific conditions: pH, ionic conditions, temperature, all kinds of things. If you stray away from those specific conditions, that protein will lose function and, lose structure and eventually lose function. So let's take a look at some data here. On the y-axis, we've got the rate of a reaction. On the x-axis, we have our condition that we're messing around with, in this case, temperature. So what we're doing is we're taking two enzymes, a enzyme from a human, right, that lives at 37 degrees Celsius, or in our body temperature, 37 degrees Celsius, and then an enzyme for a thermophilic bacteria, so it lives at 77 degrees Celsius. And what we did is we ran the reaction that's catalyzed by this enzyme in several different temperatures and measured the rate of the reaction. So as you can see, as we increase the temperature, our human enzyme increases in rate, the reaction catalyzed by that increases in rate, until we get the highest rate that, no coincidence, is right there at 37 degrees Celsius. If we keep increasing the rate of the temperature, suddenly now our other enzyme begins to increase in in activity until we get the optimal or the highest rate for this reaction right here. No, it's no coincidence that that highest rate is right there at about 77 degrees Celsius, the temperature in which that enzyme evolved. Notice that if you stray away from these optimal temperatures for either enzyme, you're going to lose activity. Why? Because eventually those enzymes will denature. And when they denature, when they lose structure, they lose function. Same holds true for pHs. We've got two enzymes, both in humans, but one that has evolved in the stomach, which has a very low pH, and one that evolved in the intestines, which has a higher pH. Again, we're going to take these enzymes, we're going to use them to catalyze reactions at different pHs and measure the rate of the reaction at each pH and plot them out on this little graph here. So as we increase the pH, going from 0 to 1 to 2, we see a higher and higher activity for pepsin until it reaches its highest activity right here, right about 2. It's about the pH of the stomach. But notice we haven't even, at these low pHs, our trypsin hasn't even become active yet. It's not until around pH 6 where it starts to become active, reaches a peak activity right around pH 8. No coincidence, that's about the pH of the intestines. So, enzymes have evolved at specific temperatures and to be active and structurally sound at optimal pHs. If you stray from those temperatures or those pHs, you're going to lose structure and therefore lose activity. Now, I want you to go back and watch that video that demonstrates how pHs can mess up the structure of proteins, because that is exactly what's going on when you change the pH of the solution in which an enzyme is acting, and that's how you mess up the function of that enzyme. Okay. All right. So after watching this video, hopefully a few times, you will be able to answer these questions. What determines where the equilibrium of a reaction be? What determines how fast a reaction will reach that equilibrium? What do enzymes do and how? What can enzymes not do? Why is heat a poor way for cells to increase reaction rates? And finally, what happens when an enzyme is no longer in their optimal conditions?